Hi there, and welcome to Gold Academy. Now, for today, we're looking at a topic in biology titled Cell and its Environment. In today's video lesson, we'll be considering the meaning of cell, environment of a cell, the processes or the mechanisms of exchange between the cell and its environment, which include diffusion, osmosis, and active transport. We'll be looking at the effects or conditions resulting from osmosis, which includes plasmolysis, haemolysis, crenation, turgidity, and flaccidity. Now, I would like you to stay to the end of this video lesson, as there are much more to learn from this video lesson. So I would implore you to stay to the end of this video. Now, let's start. The first thing we're having is the meaning of cell. Now, as we all know, cell is the basic and structural unit of life. Cell is the basic and structural unit of life. It means that all living things are composed or made up of or from cells. For example, we have a male gamete, which is the spermatozoa, and the female gamete, referred to as the ovum. This is the sex cell for the male, and this is the sex cell for the female. Now, when these two cells combine, a cell is formed, referred to as the zygote. So, zygote is a kind of fertilized cell from the spermatozoa and the ovum. So from this zygote, which is a cell, from the cell, we have the single fertilized cell, which is referred to as the zygote, undergoing what we call cell differentiation. It means that one cell becomes two, and those two, one becomes two, that one becomes two. Like, for instance, we have one cell undergoing cell differentiation to become two cells. We have this undergoing cell differentiation. This one cell becomes two, and this one cell also becomes two. So we have this. Also, each cell also undergoes cell differentiation. This one becomes two, so we have two. So we have two. From this one here, this one also becomes two, so we have this one. This one also becomes two, so we have this. This one also becomes two, so we have this. So it goes on and on and on and on and on. This cell differentiate, or we say they multiplies. So when they multiplies, they form what we call the tissue. So from the cell, we have the tissue. So this tissue are the 1,000 forms of the cell. From the cell, or let me say this tissue, let me refer you to as blood. From the blood, we have the organs forming: the hand, the leg, the mouth, the teeth, the nose. They all form. So we have the organs formed. From the organs, we have some kind of organs coming together, combining to form what we refer to as system. For instance, now, for the digestive system, we have the mouth. When you chew the food, it goes to the esophagus. From the esophagus, it goes to the stomach. From the stomach, it undergoes further digestion to the small intestine, where the complete digestion takes place. From the small intestine, the nutrients are being taken by the blood to various parts of our body. Now, the unused food that we take in are being sent to the large intestine. The ones that are needed are being taken from the small intestine, and the remaining ones, the leftover, are being sent to the large intestine, where water is being mixed with it and other chemicals from the body is being mixed with it, and it forms the feces. From the large intestine, it is being sent to the rectum. From the rectum, it's being sent to the anus, where it is being transported outside of the body. So we have different organs combining to form a system. Now, I've explained digestive system. We have what we call the respiratory system. Now, for the respiratory system, the organs include our nostrils. From the nostrils, the air goes from the nostrils, then to the trachea. From the trachea, it goes to the bronchi. From the bronchi, it goes to the lungs. And inside the lungs, we have the alveoli and the bronchioles. So these are organs that combine to form every kind of system. We have the circulatory system, which comprises of the blood, the veins, the capillaries, and the arteries. So every kind of organs combine together to form what we refer to as system. So from the organ, we have the system. We have the system. Different kind of system. So from this, when the system is completed, we have the full organism. We have the full organism. The full organism, or the baby. So it goes from the cell down to the full organism. So we refer cell to as the basic and the structural unit of life. Now, the study of cell is referred to as cytology. The study of cell is what we refer to as cytology. Now, before I clean this up, I should have explained something. Now, there is how twins are being formed. Now, I explained earlier that one zygote undergoes cell differentiation. One zygote undergoes cell differentiation to form two other two, and that other two becomes 4. That 4 becomes 8. 8 becomes 16. 16 becomes 32. 32 becomes 64, and it goes on and on and on and on until the tissue, as the blood tissue, the tissue, which is the blood, is being formed. Now, we can have an instance where we can have one cell dividing into 2, and those 2 cells, which are the fertilized cells or the zygote, can undergo cell differentiation differently. So if they undergo cell differentiation differently, then twins is being formed. If one zygote differentiates into 3 or 4, and those 4 or those 3 undergo cell differentiation differently, triplets or 4 babies are being formed. Is that clear now? So that is that about that. Now, we have discussed the meaning of cell and the study of cell. We refer the study of cell to as cytology. Now, we have the environment of a cell. Now, environment of a cell can include both the external environment of the cell, known as the extracellular environment, or the internal environment of the cell, or the intracellular environment. Now, there are differences between the concentration of the extracellular environment and the concentration of the intracellular environment. These differences is what gave birth to the processes or the mechanisms of exchange between the cell and its environment. From here is where we now have what we refer to as diffusion and the osmosis. Now, diffusion, what is diffusion? Diffusion is simply the movement of molecules from the region of higher concentration to the region of lower concentration. That is just diffusion. It's the movement of molecules from the region of higher concentration to the region of lower concentration. And diffusion takes place both in living and non-living things. Diffusion takes place both in living and non-living things. For instance now, we can have our mother cooking in the kitchen while we are sitting down at the parlor. From the kitchen, the aroma of the food, of that particular food, gets to us in the sitting room. So what happens is that there is diffusion. The aroma from the kitchen diffuses to where we are sitting down. Another instance is when someone puts on a perfume. And even though we are seated far from the person, the smell or the scent of the perfume gets to where we are. So what happens then? There is diffusion. The scent of the perfume diffuses to where we are seated. Now, another example of diffusion is when we pour, let me say, those Eve powder inside water. What happens is that Eve powder diffuses in the water and circulates around the water. So that is diffusion. So it means diffusion includes solid. Diffusion takes place in solid particles. It takes place with liquid particles or molecules. Diffusion takes place with gaseous molecules or particles. That is diffusion. Now, we have some factors that affect diffusion. Example of them, we have the surface area, we have temperature, we have concentration, we have size of the particles. For instance now, hydrogen gas will diffuse much more faster than oxygen gas because hydrogen gas is much lighter than oxygen gas. The molecular mass of hydrogen is 2 grams per mole, while the molecular mass of oxygen is 16 grams per mole. For oxygen molecule, the atomic mass of oxygen is what? Is 16. Is 16. But oxygen exists as a diatomic molecule. That would be O2. So it means the molar mass of oxygen is 32 grams per mole, while the molar mass of hydrogen is 2 grams per mole. So it means that hydrogen will diffuse much more faster than oxygen. And if you can remember in your chemistry, we have what we refer to as Graham's law of diffusion, where the law states that the rate of diffusion of a gas is inversely proportional to the square root of its molecular mass or density. And what the law is telling us is that a gas with a smaller mass will diffuse much more faster than a gas with a bigger mass. Alright, that is for diffusion. Now we have osmosis. What is osmosis? Now I'm going to give more than one definition for osmosis in this video lesson. Now what is osmosis? Osmosis is the movement of water molecules from the region of higher concentration to the region of lower concentration through a selectively permeable membrane. Now note, I said movement of water molecules. It means osmosis is pertaining only to water molecules. And I said it moves from the region of higher concentration to the region of lower concentration through a selectively permeable membrane. Now we have what we call permeable membrane, we have semi-permeable membrane, we have non-permeable membrane. For example, now a fish net is a permeable membrane. Why? It can allow anything to enter into it. That is a permeable membrane. A permeable membrane can allow anything to enter into it. Now we have the non-permeable membrane. That non-permeable membrane does not allow any particle to penetrate or enter through it. Example, we have glass. Glass is a non-permeable membrane. Now the third one, we have a semi-permeable or selectively permeable. It means those ones select what can enter into it and what can go out through it. An example of a semi-permeable membrane or a selectively permeable membrane is our cell membrane. Our cell membrane. The cell membrane can also be referred to as the nuclear membrane. That is the outer part of the cell that allows particles to come in and particles to go out through the plasmodesmata. Through the plasmodesmata, particles goes in and comes in and goes out through the plasmodesmata of the cell membrane or the nuclear membrane. So our cell membrane or the nuclear membrane is an example of a selectively permeable membrane because it does not allow any particles or every particles to go out and come inside of it. For instance, our cell membrane will never allow the organelles in the cell to move out or come inside the cell. It will never allow the ribosome to move out. It will never allow the peroxisome to move out. It will never allow the lysosome to move out. It will never allow the mitochondria to move out. It will never allow the Golgi bodies to move out. It will never allow the nucleus to move out. It only allows oxygen. It allows carbon dioxide. It allows urea. It allows potassium ion. Potassium ion is the main intracellular ion inside the cell. Sodium ion is the main extracellular ion outside the cell. So we have carbon dioxide. It allows carbon dioxide. It allows glucose. It allows protein. So our cell membrane is an example of a selectively or semi-permeable membrane because it selects what goes in and comes out of the cell. It selects it. It does not allow all like a permeable membrane. Alright, so we have defined osmosis to be different from diffusion in terms of water molecules. For diffusion, it can involve solid molecules, it can involve liquid molecules, it can involve gaseous molecules. Why osmosis involves only movement of water molecules, and it is always through a selectively permeable membrane. That's the difference. All right. Now, from this, let me explain something. Osmosis are into two types. We have the endosmosis, and the exosmosis. That is the exosmosis. Now we have osmosis as endosmosis, and we have osmosis as exosmosis. Now we have these two. Now, what is endosmosis and what is exosmosis? Now, the word endo for a biology student is not a new thing. The word exo for a biology student and a chemistry student is not a new thing. Because in biology, we have what we call endoskeleton, meaning the skeleton that is found within the body. And we have mostly vertebrate animals that possess endoskeleton. Like the mammals has the endoskeleton, the reptiles has endoskeleton, the amphibians have endoskeleton, the aves, which are the birds, possess endoskeleton, the Pisces, which are the fishes, have endoskeleton. Now we have the exoskeleton possessed by mainly the invertebrates, like crabs possess exoskeleton, that is outer skeleton. Exoskeleton means outer skeleton. Like the prawn has exoskeleton. Ants has exo, meaning outer skeleton. Now, in chemistry, we have what we call the endothermic reaction, meaning a reaction that takes in heat from the surrounding. We have what we call exothermic reaction, meaning a reaction that gives out heat to the surrounding. All right. In the term of osmosis, we have endosmosis and we have exosmosis. All right. Assuming we have this kind of medium, let me say we have this kind of medium, and we have a cell present, and we have water molecules. We have water molecules right here. Now, if water molecules should move inside the cell, we have what we refer to as the endosmosis taking place, meaning inward movement of water molecules. And remember, the word osmosis means that it's moving, it must always move from the region of higher concentration to the region of lower concentration. All right. So if we have water molecules moving from the region of higher concentration to the region of lower concentration, that is inside the cell, we have endosmosis taking place. I will soon explain why it happens that way. That will be the next thing we are going into. Now, if we have water molecules moving out from the cell, if we have more concentration of water molecules in the cell, and we have lower concentration of water molecules outside the cell, what will happen is that the cell will give out water to the surrounding. And if water is being released from the cell to the surrounding, what happens is that exosmosis has taken place, meaning outward movement of water molecules. Inward movement of water molecules is endosmosis. Outward movement of water molecules is exosmosis. Alright, with that being explained, now we have the second definition of osmosis. We can define osmosis also as the movement of water molecules from the region of low osmotic pressure to the region of high osmotic pressure through a selectively or semi-permeable membrane. I repeat, we can define osmosis again as the movement of water molecules from the region of low osmotic pressure to the region of high osmotic pressure through a selectively or semi-permeable membrane. Now, what is osmotic pressure? Osmotic pressure is caused when water molecules moves into an hypertonic solution. What did I say? Osmotic pressure is caused when water molecules moves into an hypertonic solution. Now, what is an hypertonic solution? Now, let me explain what hypertonic solution means. Remember, I said that osmosis can be defined as the movement of water molecules from low osmotic pressure from the region of low osmotic pressure to the region of high osmotic pressure through a semi-permeable membrane. And I said osmotic pressure is caused when water molecules moves into an hypertonic solution. Please take note. Osmotic pressure is caused when water molecules moves into an hypertonic solution. Take note. Alright, we have solutes plus solvents to form solution. We have solutes plus solvent to form solution. And this one is a very wonderful topic in chemistry, where we talk about solubility. And solubility, we define solvents, we define solutes, and we define solution. Solvent is just what we use to dissolve solutes. For instance now, if we dissolve salt inside water, we have solution being formed. The solute there is the salt that is being dissolved. The solvent is the water that is being used to dissolve the salt. So the solvent is the water, the solute is the salt. So we refer water to as a universal solvent. We refer water to as a what? Universal solvent, because it tends to dissolve more solutes than other solvents. Alright, we have solutes, we have solvents to give us solution. And I've mentioned some solutes that are present outside the cell. We have like sodium ion, we have like chloride ion, we have like calcium ion, we have like proteins. These are solutes outside the cell. We have hormones. They are present outside the cell. There are solutes present outside the cell. We have solutes inside the cell. We have the potassium ion. We have the phosphate ion. We have glucose. We have oxygen. We have protein. We have amino acid. We have nucleic acids. such as the DNA and the RNA. We have the adenosine triphosphate, which is the ATP. Those solutes are present inside the cell. They are intracellular solutes. Alright, when we have those solutes with water, we have solution being formed. Alright, solutes and solvent give us solution. Now we have what we call the hypertonic solution. Hypertonic solution. We also have what we call the hypotonic solution. Hypotonic solution. We also have what we call the isotonic solution. Isotonic solution. Now, hypertonic solution is when a solution contains more solutes and less water. More solutes, let me say less solvent, less solvent. Hypotonic solution is when a solution contains less solute, more solvent. Let me say less solute but more solvent. That is hypotonic solution. Isotonic solution is when a solution contains moderate, let me say the same amount of solute and solvent. The same amount of solute and solvent. So you can see that the solution is at equilibrium, is balanced. Alright, we said osmotic pressure is caused when water molecules moves into an hypertonic solution. For example now, we can have a container. Let's say we have solvent here containing less solute. And we have solvent here containing more solute. Now let me add this to it. Let me add this to it. The more the solute, the higher the osmotic pressure. I repeat, the more the solute, the higher the osmotic pressure. And the lower the solute, or the lesser the solute, the lesser the osmotic pressure. Remember what I said. I said osmosis is when water molecules moves from a region of low osmotic pressure to a region of high osmotic pressure through a semi-permeable membrane. Take note of that. Take note. Now, I said that where you have less solute, you have what? Less or low osmotic pressure. So here we have low osmotic pressure. And since there are more solutes here we contain high osmotic pressure. Here we have high osmotic pressure. Now listen very, very attentively. This place, as we all know, let me say we have the same amount of solvent here. Now, as we all know that this place contains more solute, contains more solvent than this side. I repeat, this side here contains more solvents than this side here. For example, you have a cup, and you throw, and the water level is like this. Water level is like this. Let me say they're having the same water level. Now, there is only 1 cube sugar in this one. Just 1 cube sugar in this one. And in this one, there are more cube sugar. Now, from here, you can deduce that this water level rose to this level due to the what? The more solute present. So you can say that here contains less solvent than here. The same way also, this place contains more solvent than this side here. So this one contains more solvent than this one. So you can say that this one contains more solvent or more water molecules than this one here. I repeat, this one contains more water molecules than this one here. This one contains less solute, so it has low osmotic pressure. This one contains more solute, so it has high osmotic pressure. There are more water molecules. There are more water molecules here. There are less water molecules here. Remember, osmosis is the movement of what? Water molecules from the region of higher concentration to the region of lower concentration. So there is more concentration of water here. So what will happen? Water molecules will move from this place, which is the region of higher concentration, to where there's less water molecules, which is the region of lower concentration. So as we can see, water molecules from the first definition of osmosis, water molecules is moving from the region of higher concentration to a region of lower concentration. So the first definition and the second definition are the same thing. It's just the terms used that are different. Here has more water molecules. So here has more water molecules, less solute. So it has what? Low osmotic pressure. Here this place has less water molecules. So it has less water molecules and contains more solutes. So it has what there? High osmotic pressure. Here, less solute, low osmotic pressure. Much solute, or let me say high amount of solute. So you have what there? High osmotic pressure. So water molecules is much here, water molecules is less here. So you can say that osmosis is movement of water molecules from the region of low osmotic pressure to the region of high osmotic pressure. It is same as me saying that water molecules is moving from the region of higher concentration to the region of lower concentration. So the first definition and the second definition are the same thing. Alright, we can define osmosis again as the movement of water molecules from the region of low concentration of solutes to the region of high concentration of solutes. I repeat, the third definition of osmosis is that osmosis is the movement of water molecules from the region of low concentration of solutes to the region of high concentration of solutes. That is the third definition of osmosis. Alright, with that being done, we now move to the effects or conditions resulting from osmosis. Remember, I told you that if water molecules move inside a cell, it is endosmosis. If water molecules moves outside of a cell, it is exosmosis. Alright, we have effects or conditions resulting from osmosis. We have plasmolysis, we have haemolysis, we have crenation, we have turgidity, and we have flaccidity. Now, what is plasmolysis? Plasmolysis is just the outward movement of water molecules from a plant cell when placed in an hypertonic solution. Calm down, let me say it again. I said that plasmolysis is the movement of water molecules outside of a plant cell when placed in an hypertonic solution. Listen, an hypertonic solution is what? It's a solution that contains what? High amount of solutes. So it means that when we have a cell having higher concentration of water, and that cell is being placed in an hypertonic solution where it is containing low concentration of water, what happens is that plant cell tends to lose its water to the region where there is less amount of water. So what happened there? Is that exosmosis or endosmosis? Remember, water molecules is moving from the plant cell to the hypertonic solution where there is less water. So what happened there? It is what? Outward movement of water. So it is what? Exosmosis. That is exosmosis. That is what? Exosmosis. So we can say that plasmolysis is the shrinkage of the vacuole of plants and the pulling away of the cytoplasmic linings of the cell wall of plants when they are placed in hypertonic solution. When you place a plant cell in an hypertonic solution, what happens is that the vacuole containing the water molecules shrinks, and as we all know, the vacuole or the food vacuole of a plant is where nutrients are being stored. So when water is being, the food vacuole is where water is being stored and other nutrients. So when water molecules is being released to the outside environment or to the hypertonic solution, what happens? The vacuole shrinks. Is that clear now? So that is plasmolysis. It is when water molecules moves from the plant cell to the outside environment when placed in an hypertonic solution. It tends to release water to the hypertonic solution because the hypertonic solution contains less water molecules. Alright, the next one we have haemolysis. Now, let me say this before I go to haemolysis. Take note, plasmolysis occurs only in plant cells. Plasmolysis occurs only in plant cells. Alright, next one we have haemolysis. Now, haemolysis is the bursting of the red blood cell when placed in an hypotonic solution. I repeat, haemolysis is the bursting of the red blood cell when placed in an hypotonic solution. Now, I explain hypotonic solution as a solution that contains more solvent, less solutes. So when a red blood cell is placed in an hypotonic solution, that is a solution that contains less amount of solute but high amount of solvent. When red blood cell finds itself in an hypotonic solution, what happens is that since there are much water in the environment, red blood cell takes in water. So if red blood cells should take in water when placed in hypotonic solution, what happens is that the red blood cell continues to starts to swell. The red blood cell starts to swell. If it continues to take in more water, a time will come that it will burst. So we refer hemolysis to as the bursting of red blood cell when placed in an hypotonic solution. Hemolysis is the bursting of the red blood cell when placed in an hypotonic solution. Now let me ask you, hemolysis has to do with red blood cell taking in water. Is that not so? Now, is that endosmosis or exosmosis? That is what? Endosmosis, because water molecules is being taken in from the surrounding, from the external environment of the cell. That is what? Endosmosis, because water molecules is being taken in, is being absorbed in. That is what? Endosmosis. Inward movement of water molecules. That is what? Endosmosis. Alright. The next one after hemolysis, we have crenation. Crenation is the opposite of hemolysis. In crenation is the shrinkage of the red blood cell when placed in an hypertonic solution. Crenation is simply just the shrinkage of the red blood cell when placed in an hypertonic solution. Now, as you can see that crenation and plasmolysis are somehow similar in terms of giving out water to the environment. But for plasmolysis, it occurs in plants. For crenation, it occurs in red blood cell. Alright, that is crenation. Now, for crenation, it gives out water because it is placed in a solution, because it found itself in a solution where there is less water. So what happens is that it tends to give out water to the surrounding because it found itself in a surrounding where there is less water concentration. So what happens is that it gives out the water molecules it's having inside the cell. So the outward movement of water molecules is referred to as what? That is what? Exosmosis. Exosmosis. So exosmosis happens for what do you call it? Crenation. Exosmosis happens for crenation. Endosmosis happens for hemolysis. And also exosmosis happens for plasmolysis. This is when cells give out water to the environment. Give out water. Exosmosis. Taking water. Endosmosis. Give out water. That is what? Exosmosis. This is give out water molecules from plants, from plant cells. This one is what? Taking in water molecules by red blood cells or the erythrocytes. We call the red blood cell also as the erythrocytes. This one is also giving us water, giving out water molecules by red blood cells. So that is this. Now we have the next thing. The next one we are having is turgidity. Now what is turgidity? Turgidity is the taking in of water molecules by plant cell to form what we call the turgor pressure or the turgor pressure. Turgidity is when plant cell take in water molecules to form what we call the turgor pressure. It helps plants to maintain their position, for them to be upright. Let me use that word, upright. So turgidity helps plants to be upright. When plant cell take in water, the cell membrane move itself over the cell wall. So what happens? It helps the plant to be firm, to stand firm. But if red blood cell should try it, what happens is that it will burst. If red blood cell taking water, it will burst. Why? It has no cell wall. Animal cell has no cell wall. So plant cell has cell wall. So the cell wall of the plant prevents the plant cell from bursting. Rather, when plants are taking water, it helps them to become rigid. Alright, that is for turgidity. Lastly, we have flaccidity. Flaccidity is just the same thing. Flaccidity is just the outward movement of water molecules much faster than it can absorb. So the difference between flaccidity and plasmolysis is the condition in which the both take place. For plasmolysis is that it occurs when placed in an hypertonic solution. But for flaccidity, it occurs during drought or during dryness. When the land is dry, what happens? The plants give us water through the roots to the soil. So we can curb flaccidity by watering the plants. So what happens? So that it can become turgid once again. So the condition for flaccidity is that it occurs during dryness. The condition for plasmolysis is that it occurs when placed in an hypertonic solution. So that is all for cell and its environment. If you have any question, put it down in the comment section, and where you want any, let me say, more explanation, put it down in the comment section. And also, if there's any addition, you can put it down also in the comment section. Now, if you find this video helpful, do well to like, drop a comment, hit the subscribe button, and follow us for more videos, for more helpful videos as this. Do well to share the videos also to your friends and those who are needful of knowledge in biology. Thank you. This is Ola Gold Academy.